Welcome to this explainer. Today, we're cutting straight through the buzzwords to break down the hard, verifiable biochemistry behind medical ozone. There is so much noise out there on this topic, but we're going to focus purely on the science, taking a really close look at exactly how ozone acts as a highly precise biochemical tool inside the human body. Let's get right into it.
Here's how we'll break this down today. First, we'll look at the chemistry of ozone. Then, move on to the secondary messengers. Third, we'll explore its selective targeting mechanism, followed by destroying microbes and viruses. And finally, we'll wrap up with formulations in clinical practice.
All right, starting off with part 1, the chemistry of ozone.
Let me kick things off with a question that creates a really fascinating paradox. When we think of ozone, we usually picture this powerful, aggressive oxygen, right? So what do you think happens in the very first millisecond ozone actually makes contact with biological fluids like human blood or plasma?
Well, the surprising answer is that a massive chunk of it instantly loses its oxidizing capacity and, quite literally, just disappears. It doesn't go roaming around the bloodstream like a wrecking ball. Instead, the ozone molecule itself vanishes almost immediately upon contact.
Understanding why this happens is super fascinating. It disappears because the ozone is immediately quenched by a rich, diverse army of endogenous antioxidants floating around in our plasma. We're talking about molecules like glutathione, which you can see beautifully structured right here, as well as sulfhydryls, uric acid, and vitamin C and E. They basically act like a giant biochemical sponge, absorbing that initial oxidative hit. But wait, if the ozone itself vanishes so fast, how does it actually do anything?
That brings us to part 2, the secondary messengers.
So, that initial quenching reaction we just talked about, it doesn't just stop there. It creates something entirely new. It generates reactive oxygen species, or ROS, like hydrogen peroxide, alongside lipid ozonation products, known as LOPs. Think of these guys as the true effectors of the whole process. They are the biochemical delegates carrying out ozone's mission. So while the original ozone is long gone, these secondary messengers step up to trigger a whole series of complex biochemical cascades throughout the body. And this brilliantly illustrates the concept of hormesis. If you're not familiar, hormesis is the idea that a low dose of a stressor can actually provoke a beneficial adaptive response. The net response and the potential correction of pathological states in the body depends entirely on this delicate balancing act. It's a precisely controlled oxidative challenge on one side of the scale being met by our own robust antioxidant defenses on the other. But this brings us to a really crucial point. If this process creates an oxidative challenge, why does it kill pathogens but totally spare our own human tissue?
Well, let's look at part 3, the selective targeting mechanism.
This is where the absolute genius of the biochemistry really shines. I want you to think of human cells as heavily fortified castles. They're protected by multiple, thick layers of robust antioxidant armor, rapid DNA repair mechanisms, and complex multicellular coordination. Now contrast that with single-celled pathogens. They are basically like exposed tents. They have highly vulnerable membrane structures and incredibly limited antioxidant defenses. So, when that wave of oxidative stress from our secondary messengers hits, the castle easily absorbs and repairs the damage, but the tent? It's simply blown away. Now that we know who's vulnerable, let's zoom in on the microscopic battlefield.
Moving on to part 4, destroying microbes and viruses.
To truly grasp the scale of this efficacy, consider this number. Ozone, working through those messengers we just talked about, achieves a staggering 99.9% bacterial kill rate against really stubborn pathogens like E. coli and Mycobacterium tuberculosis. And you know what's most incredible about that? It pulls this off in just 4 to 20 minutes of exposure. It does this by triggering a total structural collapse. Essentially, ozone executes a highly precise 5-step siege on bacteria. First, it oxidizes the lipids in the membrane. Second, it breaks down the structural integrity of the cell wall. Third, it completely blinds the bacteria by inactivating its key metabolic enzymes. Fourth, it collapses the proton gradient, which means the bacteria's energy production systems totally fail. And finally, step 5 is lysis. The bacteria literally rupture and die.
Viruses face a similarly grim fate, but it really depends on their armor. Enveloped viruses, like herpes or hepatitis C, wear a lipid-rich coat. And because ozone's secondary messengers are exceptionally good at lipid peroxidation, these viruses are highly sensitive and they get destroyed rapidly. Non-enveloped viruses are a little bit tougher since they don't have that lipid coat. But make no mistake, they still succumb just a bit slower via direct protein oxidation and disruption of their viral capsid.
Now, under an electron microscope, we can actually watch this destruction happen visually. When dealing with fungi like Candida albicans, you can see how the oxidative stress creates devastating longitudinal and then transverse fissures right in the fungal structure. Within just 10 to 20 minutes, this progressive damage leads to a complete ultrastructural collapse and total organelle destruction. The pathogen is, for all intents and purposes, entirely dismantled.
And the potency doesn't stop at just standard bacteria or fungi. A mere 0.1 micrograms per milliliter is enough to inactivate highly resistant fungal spores. That is a vital metric because it really showcases this biochemical tool's ability to tackle the terrifying modern crisis of multi-drug-resistant strains. It offers a destruction mechanism that these pathogens simply haven't evolved to outsmart yet.
Finally, let's wrap up with Part 5, Formulations in Clinical Practice. So because ozone gas is naturally unstable, in clinical practice, it's often stabilized into unsaturated vegetable oils. We measure the strength of these formulations by their peroxide value, or PV. The PV basically measures the concentration of those secondary messengers, the ozonides and hydroperoxides that get trapped in the oil. Looking at historical clinical data, a PV of roughly 2,500 to 3,000 is seen as the optimal range for fighting topical skin infections, like athlete's foot or candidal skin folds.
But formulating these clinical oils is always a bit of a trade-off, as we can see mapped out here. A 50% PV oil gives you really high stability and a much longer shelf life, but you only get moderate efficacy. A 75% PV gives you a great balanced clinical middle ground. Now, a 95% PV offers maximum antimicrobial potency, which is absolutely perfect for severe post-surgical wounds or burns, but the catch is, it requires incredibly careful cold storage to maintain its stability over time.
Alternatively, clinics don't just use oils. For immediate wound irrigation and local disinfection in really fast-paced clinical workflows, freshly prepared ozonated water is heavily utilized. It works incredibly fast on contact, rapidly breaking down bacterial biofilms. The catch with this one, though, is that it degrades super quickly in aqueous environments, so it has to be concentrated and used almost immediately to properly harness those oxygen radicals.
So if there's one crucial point to take away from this explainer, it's that medical ozone is absolutely not some mystical cure-all. It is an elegant, scientifically verifiable biochemical tool that operates through the strict rules of hormesis and oxidative stress. Which leaves us with a final, provocative thought. As antibiotic resistance continues to grow globally, totally outpacing our pharmaceutical developments, is it time we lean closer into harnessing our own robust cellular defenses to fight back and win the microscopic war? Thank you so much for joining me on this deep dive.